Here in part 5, we're going to talk about finding food and how archaea and bacteria survive stressful periods. Archaea and bacteria appear to have a nose. There's a chemical receptor array sitting at the head of bacteria. Here is a molecular drawing of that configuration. So it's a series of proteins that protrude out into the physical environment and through the membrane, cell wall, and the membrane. We actually have 3 faculty in the School of Biological Sciences who work on these chemotactic sensors and motors. One is Sandy Parkinson, and he works on the receptors. And then both Kelly Hughes and David Blair work on the motor. So we have a receptor, where is the food, and a motor driving the microorganism to its food. When we introduce a high resource environment, such as lots of sucrose into a growth media, we can define 3 distinct phases. There is a lag phase between when the food is introduced and the organ is able to find the food and begin replication. There is a log phase in which we have exponential growth in response to the high resource environment, so lots of replication takes place. And then we have a stationary phase when the organism is essentially run out of nutrients and is static, not dividing very much. We can also have a decline phase in which there are no more nutrients, cell numbers decline, and the prokaryote goes into a resting stage. This is very analogous to the general patterns of life cycles that we find in all organisms. When environmental resources become scarce, microbes, prokaryotes, develop a capsule or a cyst or a spore. We'll talk about each of those. The first land plants that came, the mosses, they can also go through a desiccation stage, or we can see spore formation. In higher plants, we find seed formation. We don't have an analogous feature in all animal systems. Also, although some animal systems are able to go into a resting stage that will be talked about when we talk about form and function in animal systems. Now, for prokaryotes, resources are not always available. There are high resource periods and low resource periods. During low resource periods, prokaryotes can form a capsule. And here you see capsules surrounding the prokaryote. Capsules play a number of roles. The first role is to keep them from drying out. So they are a barrier that keeps a moist water content on the inside of the capsule. It also produces some chemical compounds that reduce the likelihood of phagocytosis, or the microbes being consumed by a larger microorganism. We also find that some prokaryote form cysts. Here you see azotobacteria, and you see a number of cells, and as a community, they have formed a protective cell wall to survive harsh periods. Or you might find quite commonly the concept of a spore, in which we go from a vegetative cell to one in which there's a cell division that takes place, resulting in a mother cell which provides the nutrients and features that are necessary to produce a spore, which can last throughout a dry, harsh period. Now, for endospores and cysts and capsules, we can find that these structures can protect the prokaryote for days, weeks, months, years, centuries, and even up to several thousand years. So this resting period allows them to become dormant and persist through dormancy for very long time periods. When we begin to look at lifestyles, so to speak, in metabolic plasticity and genome size in bacteria, we find that some organisms can be classified as a super generalist. They have a very high metabolic diversity. And they also have a high genome size to sustain the sequences that are necessary for the high metabolic diversity. They might have as many as 9,000 genes or so. On the other hand, we can find some prokaryotes that are super specialists. They have one metabolic reaction that they can conduct, and that is their niche. They tend to have a tiny genome size, and many fewer, in some cases as few as 137 genes. So we can find quite contrast between super specialists and super generalists, looking at metabolic plasticity in archaea and bacteria. This study guide should help you. It is based on things that we talked about in today's lecture.